Today I am going to talk about the impact of melatonin supplementation on PCB. PCB means a polychlorinated biphenyl, orachlor 1254, induced neurotoxicity. What is PCB? These are the halogenated aromatic hydrocarbons with a 209 congeners and half-life is 2 to 8 years. The PCBs have used in the industrial and commercial applications because of their non-inflammability, chemical stability, high boiling point, and electrical insulating properties. These PCBs are used in different industrial and commercial applications, used in dielectric and coolant fluids, electrical insulating fluids, example transformers, capacitors, and electrical motors. And these PCBs are also used in plasticizers in paints, plastics, and rubber products, in pigments, dyes, and carbonless copy paper industries. And the PCBs also used in the stabilizing additives in the manufacture of the flexible PVC, polyvinyl chloride coating for electrical wiring. And the study shows that if once the PCBs are released into the environment, it causes the health hazards. These PCBs are called persistent organic pollutants. What is persistent organic pollutants? These are organic compounds that are resistant to environmental degradation through biological, chemical, and photolytic processes. Because of this, they bioaccumulate in the human and animal tissues and biomagnify in food chain that leads to cause the human health hazards. This production was banned by the United States Congress in 1979 and the Stockholm Convention on Persistent Organic Pollutants in 2001. According to US EPA, it causes cancer in animals, also in humans. But prior to 1979 ban, it entered the environment during their manufacture and use in the US. How it releases into the environment? Through both use and disposal. Still, some countries still using the PCBs, still released into the environment from poorly maintained hazardous waste sites and illegal improper dumping of PCB wastes and leaks and releases from electrical transformers and disposal of the consumer products into municipal or landfills not designed to handle hazardous waste. So, and burning of some wastes in municipal and industrial incinerators also cause the PCBs enter into the environment. These PCBs are absorbed through skin, lungs, and intestinal tracts. But humans are exposed to PCBs mainly through consumption of contaminated food, particularly from fish consumption. These accumulate via food chain. If the PCB is contaminated in the water, PCB contaminated water, the small fish, it absorbs the PCBs in water or by eating plankton. These PCBs stored in the body fat of the small fish. If the large fish is a small fish, the PCB transfer from the small fish to the larger fish and bioaccumulate in the fatty tissues. If the human eating the fish is a major source of the PCB accumulation. But unlike water soluble chemicals, they are not excreted. It leads to cause body accumulates PCB over years. And some study shows that it causes the cancer, immune function, it affects the immune functions, it affects the reproductive functions, endocrine, neurological effects were seen. How the PCBs cause the health hazards in human? It acts through the aryl hydrocarbon receptor like the endocrine function. These toxic manifestations are associated with the production of ROS. The PCBs induced toxic functions are associated with the production of the reactive oxygen species. These are the studies done on neurotoxic effects of PCBs. These alter the synaptic transmission and the plasticity in hippocampus of the rat. It disrupts the neuroendocrine disrupting chemicals. It disrupts the gonadotropin releasing hormone in the hypothalamus, thereby decrease the luteinizing hormone, follicle stimulating hormone from the pituitary. Then it leads to disturb the reproductive function. And it reduces the tryptophan hydroxylase activity. Tryptophan hydroxylase activity is an essential enzyme for the formation of the serotonin and melatonin and 5-hydroxytryptamine concentration in that brain regions. And it affects the hippocampal function in different ways, alters dendritic growth and actin cytoskeleton, and it modulates the activity of membrane bound ATPases by inducing the levels of free radicals in hippocampus and hypothalamus. These are the mechanism of action of PCB. Suggested mechanism published by scientists: the PCB enters into the cell. The PCB enters into the cell. It binds with the inactivated aryl hydrocarbon receptor. After the binding of PCB with the aryl hydrocarbon receptor, this compound is activated. The ligand aryl hydrocarbon receptor complex enter into the nucleus through the aryl hydrocarbon receptor nuclear translocator. After enter into the nuclear translocator, it activates the XRE, xenobiotic response element, and stimulate the regulatory genes, and it stimulates the formation of the cytochrome P450 enzymes and other proteins. This cytochrome P450 enzyme is the major source for the reactive oxygen species. How the cytochrome P450 induce the reactive oxygen species? This cytochrome P450 catalyzes the oxidation of lower chlorinated polychlorinated biphenyls. It induces the formation of the mono and dihydroxy metabolites. The dihydroxy metabolites auto-oxidize to give the semiquinone and quinone complex. Some studies shows that PCB quinone complex release the formation of the ROS. This is the production of the ROS by the PCBs. These are the total antioxidant system. In the time of cellular respiration and oxidative burst, in the time of environmental factors, the superoxide radical is released. This superoxide radical is highly toxic. This superoxide radical is converted to hydrogen peroxide by the enzyme called superoxide dismutase. If the absence of these enzymes, what happened? This superoxide radical converted to peroxynitrate. Okay. This superoxide radical converted to H2O2. This H2O2 converted to H2O by the two peroxidation enzymes called catalase and GPX. If the absence of these two antioxidant enzymes, this H2O2 converted to highly toxic hydroxyl radical. It causes the DNA damage and lipid peroxidations and oxidates the protein and alters the biological functions. I have concentrated in the two antioxidant enzymes. The SOD contain two types of enzymes. One is the copper zinc SOD and another one is the manganese SOD. And the copper zinc SOD is the most affected antioxidant enzymes during neurodegeneration. It catalyzes the dismutation of the superoxide into hydrogen peroxide. This copper zinc superoxide is mainly expressed in neurons. In neuronal cells, endogenous copper zinc SOD is normally expressed but is rapidly down-regulated after several types of acute brain insults, rendering the brain more susceptible to oxidative stress. And another one antioxidant enzyme is called glutathione peroxidase 4. It shows a unique cellular distribution in the brain compared to GPX1, and studies shows that these selenium-dependent enzymes in mammals with a pivotal role in the brain development and functions. This GPX4 protects the cortical neurons from oxidative injury and amyloid toxicity. And in fully mature adult brain, GPX4 is expressed in all neuronal cell layers and most prominently in the hippocampus. This is the localization of the copper zinc SOD and GPX4. This copper zinc SOD expressed in the cytosol and also in the nucleus. This is the localization of MN manganese SOD. It is localized in the mitochondria. And Auroclar 1254 is a commercial mixture of PCBs. We given the Auroclar 1254 to detect the impact of PCB on neurotoxicity. And this study shows us this Auroclar 1254 induces oxidative stress in ventral prostate, testes, lungs, kidney. And we have determined to see the impact of melatonin. Apart from several antioxidants, we have selected the melatonin to detect the impact of melatonin on PCB-induced neurotoxicity. What is melatonin? Melatonin is the main secretory product of the pineal gland. But interestingly, study shows that lowering circulating levels of melatonin also exaggerates the oxidative damage to tissues that are subjected to increased oxidative stress. These are the regulation of the melatonin secretion in the time of light inhibition and light stimulation through the suprachiasmatic nucleus present in the pineal gland. And the melatonin acts as different functions. How it acts as an antioxidant effect. First one is it direct free radical scavenger. In the melatonin structure, the structure contains the electron-rich aromatic indole ring. Electron-rich aromatic indole ring. In the time of oxidative stress, this electron donate the this melatonin donates the electron to the electrophilic radicals, and indirect antioxidant stimulation when stimulating the antioxidant enzymes. And the third is stimulates the synthesis of glutathione. Glutathione is the essential non-enzymatic antioxidant for the glutathione peroxidase. And another one function, it increases the efficiency of mitochondrial electron transport chain, thereby lowering electron leakage and reducing the free radical generations. Upon oxidation, melatonin converts to a number of antioxidant compounds. These are the metabolites: cyclic 3-hydroxymelatonin, N1-acetyl, N2-formyl 5-methoxykynuramine, and N1-acetyl 5-methoxykynuramine. These are the actions of melatonin in the cell. First one is the binding to plasma membrane, localized in the membrane receptors, and the second one is the binding to nuclear receptors of the orphan family. They directly bind to the nuclear receptor or receptor, and binding to the intracellular proteins such as the calmodulin, calcium binding protein, and antioxidant effects. These are the suggested mechanism how the melatonin indirectly induce antioxidant enzymes activity. When the cells exposed to oxidative stress, what happened? This formation of ROS, reactive oxygen species, in the time of ROS formation, the NF kappa B is dissociated from the inhibitory kappa B. This inhibitory kappa B is phosphorylated. This NF kappa B is translocated to the nucleus and binds to the kappa B response elements. Some kappa B response elements are situated in the promoter regions of the antioxidant enzymes. And another one, transcriptor factor ROR alpha, it stimulates the inhibitory kappa B and inhibit the translocation of the NF kappa B into the nucleus. So melatonin, it inhibits the transcription factor ROR through the calmodulin kinase and directly inhibit the ROR transcription factor. And the brain, like many other tissues, has a range of antioxidant defenses which help to maintain and balanced reductive status. These ROS are closely involved in Parkinson's disease, schizophrenia, and Alzheimer's disease. We have selected 3 brain regions for my experiment, and cerebellum, it is essential for coordination of movement and balance, and cerebral cortex, sensory information or coordinate motor output necessary for control of movement, and hippocampus, memory function, it involved in memory function in humans and other animals. And studies also shows that the cerebellum, cerebral cortex, and hippocampus are highly sensitive to oxidative stress. This is the first. In the chapter 1, we have determined the impact of melatonin on PCB induced changes in antioxidant system in all the 3 brain regions. These are the materials and methods. Group 1 is a control, group 2 is oracular 1254, this is a PCB 2 mg per kg body weight IP, and group 3 oracular 1254 2 mg per kg body weight IP with a simultaneous treatment of melatonin 5 mg per kg body weight. And group 4 is oracular 1254 2 mg per kg body weight plus simultaneous administration of melatonin 10 mg per kg body weight, and group 5 and 6 are melatonin, separate melatonin controls. The dosage and duration of PCB was selected according to our previous studies, and the dose level of melatonin was selected according to Gomez et al. And Feng and Zhang et al. These are the body weight of the animal. The body weight was significantly decreased after PCB treated animals, but it is not retrieved after the simultaneous melatonin treatment in both the doses. The rat brain was dissected to cerebellum, cerebral cortex, and hippocampus. Regions from each of the brain tissue were plotted very accurately. These are the regions, brain regions weight. The regions brain regions weight were decreased in the PCB induced animals, but it is not retrieved in the simultaneous treatment of melatonin in all the 3 regions. And enzymatic antioxidants were observed, non-enzymatic antioxidants, GSH, vitamin C, vitamin E, and reactive oxygen species, lipid peroxidation, hydrogen peroxide, and hydroxyl radical. And specifically mRNA expressions were observed. One is a copper zinc superoxide dismutase and glutathione peroxidase 4 expression. These are the non-enzymatic antioxidant. The antioxidant enzymes activity was decreased significantly in PCB treated animals, but it retrieved after the simultaneous treatment of melatonin in both the doses in all the 3 regions. The other enzymes, the same trend was observed. These are the non-enzymatic antioxidant, vitamin C reduced to glutathione. And coming to the free radicals generations, the H2O2 was significantly increased in PCB treated animals. In the simultaneous treatment of melatonin, both the treated were significantly retrieved the same, and the same was observed in hydroxyl radical and lipid peroxidations. Coming to the RT-PCR data, copper zinc SOD and GPX4. This is a mRNA expression of the copper zinc SOD in cerebellum. This is A indicates a DNA ladder, B indicates a control rats, C indicates the PCB treated rats, and D indicates the melatonin simultaneous 5 mg, and E melatonin simultaneous 10 mg and melatonin control. The band was significantly decreased in PCB treated rats when compared to the control. The same trend was observed in the GPX4. The band was significantly decreased in the PCB treated animals. The same trend was observed in all the 3 brain regions, cerebral cortex, hippocampus. Coming to the chapter 2, we have observed the effect of melatonin on PCB induced changes in membrane bound ATPases, Na plus K plus ATPases, magnesium ATPases, and calcium dependent ATPase, and creatine kinase system also. And the creatine kinase system, serum and tissue CK isoforms were separated using polyacclimate gel electrophoresis, along with the acetylcholine esterase also estimated. The membrane-bound ATPases was significantly decreased in PCB-treated animals. After the simultaneous administration of both the melatonin treatment in both doses, it retrieved the same. Coming to the creatine kinase, and creatine kinase is used as a reliable marker in the assessment of myocardial, muscular and cerebral damage. There are 3 isoforms in the creatine kinase. Yesterday I told. One is the brain specific, cardiac specific and muscle specific. But in the brain specific plays a key role in regulation of ATP level in neuronal cells. In the data, the creatine kinase was significantly decreased in the PCB treated animals because of the R-wise generation, and it was retrieved after administration of simultaneous melatonin treatment in both the doses. But in serum, creatine kinase level was increased in the PCB treated animals. Because of the brain damage, it leads to leakage of the enzyme from the brain into the circulations. This is a isoform data. The creatine kinase BB was significantly BB and the MB was significantly increased in PCB treated animals. It was retrieved after administration of melatonin treatment. These are the isoforms in brain regions. The creatine kinase level was significantly decreased in brain regions. The reason is the CKBB, creatine kinase, brain specific creatine kinase, it contains the ATP binding domain. It contains the essential amino acids such as cysteine, arginine, histidine, lysine. These are targeted by reactive oxygen species. These reactive oxygen species interact with the cysteine. It leads to cause the sulfityl groups in cysteine located in the active site of CK. It may be inactivate the enzymes. And another one reason, formation of carbonyl contains derivatives when the reactive oxygen species acts on the active site. And acetylcholine esterase was also determined. This acetylcholine esterase is a neurotransmitter, and then the activity was significantly increased in the PCB treated animals, and it was retrieved after the administration of the melatonin both the doses. Coming to chapter 2, effect of melatonin on amyloid beta protein expression in cerebellum, cerebral cortex and hippocampus of the adult male rats. Yesterday I explained. This amyloid beta protein contains in 35 residue methionine. It is methionine 35 has been shown to be a critical residue in amyloid beta induced neurotoxicity. These are the Western blotting data, the primary and secondary antibodies purchased from the Santa Cruz. The amyloid beta level was significantly increased in PCB treated animals because of the ROS induces the proteolytic cleavage of the APP to give the beta amyloid accumulation. And in the simultaneous administration of melatonin in the both the doses retrieved the same. The same trend was observed in all the 3 regions. The melatonin levels in serum and brain regions of treated animals were also observed. The serum level of melatonin was decreased because of the PCB inhibit the 5-hydroxytryptamine and tryptophan hydroxylase enzyme. This tryptophan hydroxylase enzyme is essential for the formation of the melatonin. Because of the inhibition of this enzyme, the melatonin level was decreased in PCB treated animals. And the tissue melatonin levels was also significantly decreased in all the 3 brain regions. Coming to the histomorphological changes in all the 3 brain regions, this is a control rat. In the PCB treated rats, the pygnotic perineuronal spaces and pygnotic nucleus and neuronal shrinkage were present in the PCB treated animals because of the ROS inductions. But in the melatonin in the 5 mg treated, the small amount of the degenerated neurons were observed, and in melatonin 10 mg there were no significance. The same trend was observed in the hippocampus 40x. This is a pyramidal cell layers. This is a pyramidal cell layers essential for the memory transformation. In the pyramidal cell layers, this is a control rat, non-neuronal cells. In the pyramidal cell, when compared to control, the pyramidal cell layers are degenerated. These are degenerative neurons. And in PCB, in melatonin 5 mg, the small amount of degenerated neurons were observed. Coming to the cerebellum, this is a Purkinje layer of the cerebellum region. In the cerebellum, the degenerated neurons were observed in the PCB treated animals when compared to the control rats. So, coming to the overall summary, this is a cell. This PCB entered into the cell, it binds with the aryl hydrocarbon receptor nuclear translocator. then it translocates into the nucleus by the aryl hydrocarbon receptor nuclear translocator. It activates this genobiotic response element. This genobiotic response element regulates the genes, and it induces the cytochrome P450 enzymes. This cytochrome P450 enzymes is a major source of the reactive oxygen species. This reactive oxygen species inhibit the creatine kinase activity, and it also inhibit the membrane bound ATPases, and it also inhibit the acetylcholine extrase activity, and it induces the proteolytic cleavage of APP. It induces the accumulation of the beta amyloid in the extracellular region. Because of melatonin is an antioxidant, it inhibits the oxidative stress, and thereby it retrieves all the biochemical parameters. Finally, conclusion: PCBs are neurotoxic compounds which induce the production of free radicals leading to oxidative stress. Oxidative stress and formation of free radicals are the major factors of the cytopathology of many neurodegenerative diseases. Melatonin, which possesses characteristic of an antioxidant, is a leading candidate for the prevention and treatment of neurological disorders. This study proves the usefulness of melatonin in disorders affecting the brain free radical mechanisms. And this work was published in Neuroscience Research in 2010, the mRNA data. And these are the biochemical data published in the International Journal of Developmental Neuroscience. And the creatine kinase data was published in the Basic Clinical Pharmacology and Toxicology. And this work was highly commented by the Hardelin, is a German scientist, and this work was published in the review, one of the review, published in the Current Neuropharmacology by the Russell Ritter. He is working in Department of Cellular and Structural Biology. These are my publications. These are my another publications related to PCB. Thank you.